So good afternoon for the ones that just joined us over Zoom. Hi again if you stayed for the second presentation. So thanks everyone for continuing. So it's with great pleasure that I would like to introduce Parker Persons to provide his exit seminar to our department. So you might not know, but earlier on in the process, a little over 2 years ago, Parker stepped into my office and wanted to work with me with hydroponics. So he was then lured to work with Mark, which was a great opportunity for him to do the first portion of his project. And then he joined my program, and we had the opportunity to be working together for the last 14 months. So Parker is going to bring everything that he had done. And I have to say that he matured a lot as a professional, coming straight out of undergraduate here with us in the department, and now being a very respectable professional in the company. So without further ado, folks, I would like to let Parker talk about boosting photosynthesis using perovskite solar panels in greenhouses and airflow and light levels in vertical farms.
Perfect. All right, thank you, Ronito. I appreciate it. So yeah, I'll tell you guys what I've been doing the last 2 years here at University of Georgia. So yeah, it said the title. So here's how I'm gonna go about it. I'm gonna do an introduction to the first chapter. So I guess let me back up. So both of these studies are completely different. So this is why I broke it down like this. We'll do an introduction to the first chapter, dive into the first chapter with everything included that, and then same thing for the second chapter. Oh shoot. So yeah, a little introduction to the first chapter. Agriculture is a very carbon intensive. We have a big carbon footprint in agriculture. Actually, as of 2021, 11% of the U.S. greenhouse gas emissions were from from agriculture, modern agriculture specifically. Click on the screen. Here we go. Sorry, guys. Okay, so automation of greenhouses, large carbon footprint, need high electricity needs, and this goes for vertical farms as well. So we can lower this carbon footprint. I focus on semi-transparent perovskite solar cells for use as glazing material on greenhouses. Produce energy while lowering the industry's carbon footprint. So agrivoltaics is a big term for this section of research. So it's solar energy basically in tandem with crop production. So as you can see here in this picture, this is a very intensive picture to show, but yeah, so solar panels basically just providing energy for all of the electrical needs for the greenhouses, and agri relating to food, voltaic energy production. So the common solar cell is a crystalline silicon solar panel, which you see on houses or universities or any businesses, and these are completely opaque, have a high efficiency, low cost, and a long lifespan. But and that's the majority of the solar cells in the U.S. and throughout the world as well. But there's been a lot of research with physicians or physics departments and everything around the world about perovskite solar cells. So this is a picture of it up in the right corner, and they can be semi-transparent. So unlike silicon, crystalline silicon perovskite is not a single material like that. It's a family of compounds that has the same crystalline structure. So you can have a backbone and add things onto the end of it to create more efficiency, create different optical properties and whatnot. So they're cheap, lightweight, and more efficient than your crystalline silicon solar panels that you see the majority of places. So this is why there's a lot of research going on with these panels right now. So and lead halide perovskites are that backbone that is being researched a lot right now. So the semi-transparent nature of them, they can have wide range of optical properties and allowing for that transparency like I showed back here. And smaller concentrations, thin layers of it, they still produce very efficient energy production with even low molar concentration and thin layers of these across the substrate. So just an example, they are deployed even as windows in large buildings now, they're starting to be. And so these produce energy while also shading the building, so that would be even less energy produced for the building or needed to be produced for the building because to cool it off based on the sun's coming through the windows. So I propose, why not greenhouse too? There's some buzz right now about using this as a glazing material on greenhouses, these perovskite solar cells. So chapter 1, the title is Greenhouse Applications of these Semi-Transparent Perovskite Solar Cells. Knowledge gaps. There's been a lot of, or there has not been a lot of research done even using these as a greenhouse glazing material or looking at plant responses of perovskite solar cells or of plants underneath perovskite solar cells. So, and specifically, I wanted to focus on the photosynthetic response of these crops, like lettuce specifically, underneath these perovskite solar cells to see if there is a good fit for it in the greenhouse industry. The objectives would explore these perovskite solar cells as a glazing material and possible potential benefits, obviously electricity production. It could be a great use as a shade cloth substitute as well. Don't spend money and use an unrenewable resource for shade cloth. And can it enhance crop growth? There's actually been some companies that have the glazing materials on greenhouses that change the spectrum of light when it hits it, and it can enhance crop growth. So we wanted to see what happens on the photosynthetic response of when using perovskite solar cells. So this is materials and methods now of the first study. So I did 2 trials, and these were the environmental conditions. And to the picture here will show so all the crops are grown in a controlled environment before being taken to an experimental section, which I'll show you in a second. But kept both environmental conditions about 23.5 degrees Celsius, around 800 ppm of CO2, and around 1.1 kilopascal vapor pressure deficit under a 16-hour photo period and 250 micromoles per meter squared per second light intensity. So on the left here, you will see this is trial 1 solar panel or like solar cells that were given us by the physics department, and then this is on the right the different compounds that they gave us for testing. So so this is it's a little complicated, so I want to show some pictures of this experiment. So I have 2 high-powered LED lights above and on a pulley system, as you can see all the way to the right. And once they were mature enough, the plants, we took them out of the growth chamber, brought them to this experimental section, and used the CYRS-3 gas exchange analyzer to find the gas exchange properties of the plants underneath these perovskite solar cells. So what I did was I started at the first trial. I started at the lowest possible that the dimmer would let me, which was around 20 ppfd, which is barely anything for plants. And then I doubled it each time, the light intensity, until I reached around 16 to 1800 ppfd. And that's where the pulley system comes into play right here. So that was a lot of fun. I had to pull the lights down on top of the plant to get those highlight readings. And I would switch out each treatments for the perovskite cells, switch them out on top of the gas exchange analyzer each time. So the hypothesis was that the assimilation the maximum net assimilation rate and maximum quantum yield of photosynthesis would be greater with the control, which is just 2 glass slides, than the perovskite cells. So in trial 1, we used different concentrations of a methylammonium lead iodide perovskite. And so these are the 3 concentrations, which is 0.06, 0.12, and 0.24. So you can see they get darker in the picture at the bottom right. And a control as well. So we use the CYRS-3 gas exchange analyzer. From this, we created light response curves, and with all the data and for each treatment, and that was a non-parametric 3-parameter nonlinear regression curve. And from the equation, we were able to find the maximum quantum yield of photosynthesis, maximum net photosynthetic rate as well. And the transmittance was also tested of light going through each of these panels with a spectroradiometer. We'll start with the transmittance of the 3 different concentrations. So as we can see, the black line up here, that's the control, which has about 90% transmittance of all wavelengths through. And then you get down into the 0.24, the highest concentration of the perovskite. You can see that all the perovskites follow that same trend. It's just the concentration makes it go even further down. So you can see how they're blocking a lot of light in the blue region, and then they're letting a lot of light in through the red and far red regions. And these are the light response curves that I created from like the nonlinear regression curves for each of the treatments in 6 different replications, and should have included the R-squared values, but all of them were above a 0.97 R-squared, which is very good. And so the lines of fit, the light response curves fit very well for these. And yeah, so just to show you that there's really not a lot of difference in them, and that'll be kind of a theme going throughout this study, is that we don't want to see differences. And if we do see any differences, we would want to see, maybe even enhanced photosynthetic capacity underneath the perovskite, the glazing material. So this shows just a 95% confidence interval. I had put all the points from every replication that I did, and I put all the regression curves in there, and then I made the control 95% confidence band. You can see that almost all the points and all the regression curves are underneath there. So again, we're seeing no difference, and the control is not standing out or anything. So then we can get into the maximum quantum yield of photosynthesis. There's no statistical difference found in the maximum quantum yield of photosynthesis for any of the 3 concentrations or the control. And again, or the maximum net photosynthetic rate. So we did see a difference, a significant difference in the highest concentration, which is very interesting because maybe some of those optical properties of it, when the light had gone through, letting more light in the red region, whatnot, and then maybe how it's dimming the light and not over capacitating the photosystems. Maybe that's why we're seeing a possible higher maximum net photosynthetic rate. But the other 3 remained all non-significant from each other. So now we're going to go to trial 2. So this is where the physics department was doing a lot of tests on different efficiencies of these compounds. So like I said, the perovskites can have a backbone. And so this is a methyl ammonium lead backbone here. And then they tried iodide and bromide added onto it with different concentrations of each combinations of them. So same exact measurements as trial 1, just one difference is that I use categorical PPFD this time. I decided I only need to define 3, a low, kind of medium high, and then a high PPFD to really get that light response curve that I wanted. So here's the transmittance of all these panels. So they follow the same trend basically as the methyl ammonium lead iodide, just in different shifting up some. And as you can see, the control again letting in about 90%. So, and then just to note again that the blue region, these perovskites, they like to block in the blue region more than they let a lot more in the red and far red regions. So again, 95% confidence for this one for the control nonlinear regression curve, and you can see that from all reps and all from every rep of this experiment, you can see that all the lines or all the points fall within that. This is a great thing, like I've been saying. That's the theme of this study. So again, the maximum quantum yield of photosynthesis for this study. There's no statistical difference in any one of these perovskites, and they're all at the 0.06 molar concentration. I should have mentioned that earlier. We decided that was all we needed to find differences within the treatments. And then again here, maximum net photosynthetic rate, there was no statistical difference between any of the perovskites. And I wanted to show also at the different light levels to see what was happening at the individual light levels. So 200, 600, or 1200, they all did not have any statistical difference from the ANOVA tests. So takeaways, photosynthetic performance, like the theme I've been saying, is that these perovskites really had no difference in the photosynthetic capacity than the control cell. And so speculation as why, like I said, those optical properties when light was passing through and could let more in the red region and leave or block more in the blue region. That could be why we see this, because red and far red light like to enhance that vegetative growth, and our enhanced photosynthetic rates too more than blue light. Photosystems like it more, and there's synergistic effect of all light wavelengths with far red light. So not only do they change the wavelength, these perovskites, but I think it'd be a great addition to use to generate energy on greenhouses, and it should be explored using as a glazing material. And a great sustainable substitute to shade cloth. Blocked a lot of light, and so if these were put on greenhouses, you can even get rid of having to buy your shade cloth. So I do think there needs to be a future plant growth study with this. Unfortunately, we had to cut it a little bit short, and we wanted to get funding for large panels and do actual growth studies in greenhouses, but that was not able to be done based on funding. And so yeah, I think it'd be very cool to see what happens in a growth study in multiple seasons. And but I expect to see similar results from long-term growth study as I did from a short-term study. So some acknowledgements from this experiment. Dr. Van Ursel was very monumental in helping me design this experiment. He developed the whole thing, and we miss him very much. And members of the physics department, they were helping. They provided all the perovskite solar cells. So Gian and Dr. Ulrich, Dr. Nguyen, and Dr. Ferrarezi for helping me analyze everything and bringing this study to an end. And this was funded by the presidential seed grant. Okay, so I will move on to chapter 2 now, the introduction, and then go into chapter 2. So vertical farming, no pun intended, is a fast-growing industry. It's a great way to produce your produce, you know, in close to large cities or in large cities. So it can even cut your transportation distance of your fresh produce by 99% if you live right next door to this vertical farm. So that's great. And controlled environment agriculture, indoor, we can control all the environmental conditions there, hydroponic growing, you know, you can not use much substrate. And we can maximize yields while minimizing disease and inputs. 100 times more productive than per square foot than traditional ag. So calcium, as a plant nutrient, is a micronutrient, essential in plant for plant growth, and it's essential plant nutrient. Cation, Ca2 plus. So some of the roles that calcium plays in crop growth is a structural role, cell membrane, and the cell wall. It's a messenger, the cytosol, and activates certain enzymes within the plant. So it's very important to have sufficient calcium composition within the plant. The calcium tip burn, it really is a grower's nightmare. So it's an immobile nutrient. That means that it's not readily transported through the phloem, the nutrient pathway. It has to be transported via the xylem, and which is the water pathway. And it is, the calcium tip burn is a localized deficiency in the tip sleeve. So you can see here in the picture, you know, a lot of growers deal with calcium tip burn nowadays, and you can see here that like the tips just look like someone took a lighter and burned them, and that is, and like I said, it's an important structural component in the cell wall. So that's why when the rate of growth exceeds the rate of calcium transport, that's why the tips look like that. And so this is a big problem for growers because it causes lower yields and unmarketable products like seen here. No consumer really wants to buy any product that looks like that. And a lot of growers are actually cutting their cycle short because they don't want to deal with calcium tip burn and lose money from their production. So what's causing this? Well, in the vertical farm industry, we are trying to promote such a fast growth environment for these crops, like high CO2 supplementation, high relative humidity, vapor pressure deficit, and the high temperatures and the high light levels. The growers want to have as big of a crop, as much yield with, yeah. So that is why we supplement all this in a vertical farm. But hopefully we can find a way to minimize calcium tip burn while still doing all this. So chapter 2 is downward airflow rates on canopy versus light intensity to minimize the calcium tip burn disorder in lettuce production in vertical farms. So knowledge gap. So there has been experiments that have shown the effectiveness of downward airflow on minimizing calcium tip burn. So that is known. But the gap here is there's minimal comparison of the varying downward airflow rates. You know, a lot of experiments will hone in on one airflow rate and not compare them with any different airflow rates. And And then, yeah, and there's not really any experiment that I know of that is coupling airflow rates with varying light intensities as well. The objectives will be to grow Casey and Dragoon cultivars of lettuce in the same environmental conditions, replicating in 2 seasons. And we'll do periodic resource use data collection throughout that growth period. And we want to find optimal growth or optimal airflow rates, light levels, or the combination of both to minimize this disorder in lettuce vertical farm production. So obviously we want to make suggestions to commercial production facilities and growers. And we want to be able to have growers not fear extending their growing season to the full length without calcium tip burn coming. The cultivars used were Casey and Dragoon on both of the seasons. And the duration of the experiment, the seedlings were grown 14 days on each season in a controlled environment walk-in grow chamber we had on the University of Georgia. And rock wool plugs and about 800 ppm CO2 at 24 degrees Celsius with a 1 kilopascal vapor pressure deficit. And every 3 days they were watered with 100 ppm nitrogen fertilizer to give them that boost. So the 2 growing seasons were pretty recently, December through January, and then January through February. And just to note, Dragoon was harvested after 27 days. I wanted to do all of them on 28 days, it's just a lot of plants, and I had to split them up into 2. So 27 days Dragoon and then Casey after 28 days. The experimental setup was a split plot design in deep water culture, and it was an old storage closet turned into a vertical farm thanks to our very own Chris. And so the 2 treatments for the airflow rates and light levels. So the airflow rates were from 0.04, 0.07, or 0.7, 1.0, 1.3 meters per second. And the light levels were 200, 350, and 500 micromoles per meter square per second. And again, we wanted to go a little bit higher on the light level, but the power was the issue there. I'm trying to increase that light level. So just a little, I'll go quick through this little diagram right here. So the 2 different deep water culture trays per section. We had 1 cultivar Dragoon, 1 Casey. Then the lights above, we dialed these into the specific light level. And then above, we had an inline blower connected to an airflow poly sleeve. And the different spacing of holes and the different size of the holes was what, through an equation, was able to get us an average airflow rate for each of these between these. So this is just a visualization of it, show better. So you see how there's 3 different sections, each and three racks. Each shelf has 1 airflow rate, and then they're split, or they're cardboard like that, split by section of the cardboard to get the different light levels. And this just further shows how I randomized all the light levels, and each rack had each shelf had a different airflow rate. And then the light levels were randomized. So the environmental conditions after transplant inside the vertical farm, about 21.5 for both seasons, degrees Celsius, about 65%, give or take, relative humidity, 0.9 kilopascal vapor pressure deficit, and around 800 ppm of CO2 that we had supplemented during the photo period, which was a 16-hour photo period. And those were the varying light intensities as well. So this is a nutrient solution that we use to grow the crops in the deep water culture. And you can take a look at the chart if you would like, but the target pH we had was 5.5 through 6 pH, and then the target EC was 1.55 electroconductivity. So that was the full strength. And so what I did was, for both trials, I started with that full strength. Crops were put in. Then after 2 weeks, when the crop started using a lot more of that water, I refilled with half strength because the EC was also creeping up, and the water was evaporating, leaving the EC to increase more. And then after 3 weeks, I topped back off with full strength again to kind of just balance out that electroconductivity within the solutions. So the measurements that were taken through the nutrient solution, we did electrical conductivity, pH, temperature, and water use efficiency, which was just volume of solution loss, really. And the pre-harvest would be canopy volume, the length with height of the canopy, leaf anthocyanin, leaf chlorophyll, and gas exchange. And then the destructive measurements during harvest, we did fresh weights, dry weights of both the roots and the shoots, and then leaf area, soluble solids content, titratable acidity, tip burn intensity, tip burn ratio, and then a leaf nutrient analysis. Waters Ag Laboratories. So that was sent off to them. So the nutrient, we get into the tables of the nutrient solution and pH, EC, and temp. So we see that light intensity does affect, known to affect, pH and EC and temperature. So as we can see, like as you increase the light intensity, the pH will increase, and then when we decrease light intensity, the EC had a higher average, and that's really just because we know that in a higher light intensity, they're going to use more nutrients faster, and the growth rate is higher. And the same thing happened with the second season. So the first season on the left, second season on the right. That'll be for the rest of the charts, by the way. And so the temperature was significant on the second season as well, which is as expected. A higher light intensity would produce a higher solution temperature. So we did days after transplant was very significant because we don't have automatic pH dosers in there that we wish we did, but so about three times a week I went in and adjusted pH, measured it and adjusted it and remeasured it. So you'll see some fluctuation, but I think I did a pretty good job keeping it within the 5.5 to 6 target goal that I wanted to. And there was some airflow times light intensity interactions with the electrical conductivity for both seasons. So the same exact thing happened with Casey. They were all same solution, same growth room, so they're not going to see really any difference. So I'll go ahead and skip through these. Same airflow times light intensity treatment interaction on the electrical conductivity. So these are just treatment interactions of electrical conductivity in the solution, and we can see that the trend is that the lower light levels will have a higher average EC than the other ones. So then we'll look at the macronutrients. We sent these plant tissues off to Waters Ag Laboratory, and we want to look at light intensity first. So nitrogen in both seasons had a higher or had a lower concentration at the higher light level, and that's really just because of the dilution effect of a higher biomass with a lower, which really they probably had they have more nitrogen in it, but when they got sent back, that's why they're lower. And the calcium, interestingly, has more with a higher light level, and it probably because the higher light level you're transpiring more, so calcium will be translocated through the shoots more. And then there's some treatment combination significance here. So if we look here, the trend is the same, that dilution effect we can see with the lower light levels having more of a concentration than the higher light levels. But we can see that in the magnesium in the middle graph, and even over on the sulfur, you can see that the high light level does have a pretty high, and it's at the highest significance level of concentration, and that could just be from like the high airflow rates, making up for the high light level basically, and transporting the nutrients. Oops. And then interestingly, we will see this in the 0.7 airflow rate with the low light intensity, but and almost all these graphs I've made, that one is a lot lower than the rest, which is interesting. Then we look at the KC macronutrients, and same thing, except this time phosphorus was significant over here, and same with sulfur, and all the nutrients in the second season. On light intensity. So then the micronutrients, look at these, and here we only saw one of the single treatments of airflow was significant for copper, and you can see over here that at the highest airflow rate, it had the highest concentration of copper, and that could be telling because copper is also an immobile nutrient like calcium, and we transport it through the xylem, so So maybe the higher transpiration rates that are happening under these high airflow rates will be, relocating that nutrient throughout the plant. Another treatment interaction here with the zinc. And again, we can see that the lower light level, because that dilution effect with the biomass, is the lower light level usually will have the higher concentration of it for biomass, but then we see that in the high light level, we had a spike during the high light level with the high airflow rate. So then again, you see the 0.7, 200, 0.7 airflow with 200 light level, having that low, interestingly low concentration. So the KC micronutrients, same thing over here with light intensity, that dilution effect happening again. So then Dragoon, we're going to the biomass of Dragoon of both KC and Dragoon. So as expected, with the weights, shoot fresh weight, shoot dry weight, and the fresh weight, root dry weight, higher light levels will show a higher value for those. And same thing for both seasons. And then canopy volume, we can see that the opposite happening with the lower light level having higher values because plants when they have lower light, they will expand and try to catch as much of that light as possible, besides compacting like the higher light level plants. And then for Casey, same thing happened here. And that's as expected, nothing new. So yeah, there was an interaction in leaf area and this. Yeah, so these within the same airflow rate, we saw that the medium one had the highest airflow, but usually you can see that trend that the lower light levels have the more leaf area just because, like I said, they expand more. When we get into soluble solids content, titratable acidity, and chlorophyll and anthracene in the pigments. So for soluble solids content and both of the pigments, we see that higher light levels will have a higher value, which is known. And you have more light, more biomass, and more sugars will accumulate with soluble solids content, and then with the pigments, more light, they're trying to protect themselves and produce more of this pigment. So you will see higher pigment content there. And then for the Casey, same thing happened. And this time soluble solid content was significant on both. And then we couldn't make out what this was, but the airflow rate was significant for the titratable acidity. Now, anthracene and treatment interactions, we can see there really was no definitive from any of the airflow rates, but with higher light level, we tend to see a more accumulation of anthracene because they're trying to protect themselves from the high levels of light. And then we're getting into the tip burn intensity, which is ranked 0 through 5, which is 0, which is no, you didn't see any tip burn on it, or 5 was the most severe case. And then tip burn ratio and water use efficiency. So tip burn ratio is just the ratio of tip burn leaves to healthy leaves. And so we can see with light intensity, as expected, tip burn incident increased. They're growing way too fast, can't keep up with the calcium demand in those shoots. And then tip burn ratio, we see that with higher light level as well on both seasons. And then water use efficiency, which is labeled as water use, it is that was basically volume of water lost or used from the plants. And so we can see the higher light levels also using more water as expected. And so tip burn incident though, which is interesting to see that this is exactly what we want to see. Airflow rate is highly significant in the first season and the second season. That the highest airflow rate minimize the tip burn intensity within the plant. And then you see on the second season too, even water use efficiency, which was the amount of water used. This is telling of transpiration within the plant. So yeah, here at the higher airflow rates, they're using more water, and that's just telling that they're transpiring more. And so same thing with Casey here, and light intensity, same trends you can see here. And then here we got water use efficiency, which is water use on both of them. So that is definitively telling us that they are transpiring more, and when they're transpiring more, then more calcium is able to be moved into the tips of the shoots and prevent this calcium tip burn. Now this is just some pictures I wanted to show. I'm gonna only show Casey. If I go back real quick, you can see that if you look at the 200 in light intensity and the tip burn intensity and the tip burn ratio values, there's really none. At the lower light level, Casey does really well at not producing any calcium tip burn. So this is 200 light level from 0.4 up to 1.3 meters per second airflow, and you really can't see any tip burn here. And then we go to 350. Now you can see at the 0.4 might be hard to tell, but you're starting to see some tip burn in here, and especially if you go underneath these, because those plants now aren't getting as much airflow on their canopy inside of those closed up balls. And then yeah, as you move along, it gets less and less tip burn incidence. So then we can move to 500 ppfd. You'll see at the low airflow rate there is significant amounts of tip burn here. And then still as you move up a little bit here, you can see inside of here, and then once you get to 1 to 1.3, you really can't see any. And that was I tried to choose pictures that had best shown of what the average was. So yeah, this is just evidence to show that a higher airflow rate will minimize calcium tip burn even at the high light intensity level of 500 ppfd. So then the last measurement I took was gas exchange. And so we're going to look at net assimilation rate, transpiration rate, which is E, and then water use efficiency, which is just assimilation rate by the amount of transpiration happening. So here, as expected, with increasing light intensities, we will see an increase in net photosynthetic rate of the plants, and we'll see an increase in transpiration rates as well. They're trying to cool themselves off, so they are transporting more water and transpiring through the shoots. So and then water use efficiency. A higher water use efficiency means that there is more, or if you have a lower water use efficiency, that means that calcium is able to be transported more readily up because the transpiration rate compared with the photosynthetic rate is higher. So, and then this is what we like to see over here, the airflow rates. Yeah, and I only did it for the second season, by the way. I had compromised data for the first season. And so the Dragoons on the left, Casey is here on the right. So the airflow rate was significant in the transpiration and water use efficiency. So as you can see that the highest airflow rate produced the highest transpiration rate, meaning that it's working, that these higher airflow rates really are making them transpire more, putting calcium in that pipeline and bringing it up to the shoots. And water use efficiency, so like I said, the lower water use efficiency means that there is greater transpiration happening compared with photosynthetic rate, and that can be telling that calcium is more able to relocate to the tips of the leaves as well. And this is just a picture of how I took the measurements. I took them in real time. I had made sure that the light spectrum was the same within that LED fixture that is on top of the gas exchange analyzer. So that was fun. That was had to be done at night so some of it I finished at like 3 a.m, so that was a lot of fun to do. And you can imagine getting up to the top shelf up here and how tough that is too.
Why did it have to be done?
Well, we had done the photo period at night. So the lights were on at night, and I actually thought when the second season was about to happen, I thought to change it to the daytime to avoid this, but it's okay. I think it's better to be in there with all the lights on when you're doing your measurements during the day.
So would you do it before or after going to the bar?
In between. Yeah.
So
That's a good question.
Top.
Yeah, that's why I was exactly. Wobbly. And then the second, actually the first season, I was having to like. Ronnie don't want like to hear this, but it was dangerous for this machine. But then we found the strap to use. It like attaches to you. So some takeaways here is that we did see that increasing airflow rate really does be proved to minimize calcium tip burn. It is great to compare them with in the same study with different lower airflow rates. And it gets more complicated when we add in light intensity with it as well. But ultimately, like you saw in those pictures with Casey, it does minimize that calcium tip burn that you see there. It's just I really think that the problem is with the higher airflow rates is that the canopy gets so compact and you're blocking a lot of those leaves within there that aren't getting that downward airflow rate and able to transpire and bring that calcium up. So yeah, multiple measurements like I showed to increase transpiration rates. We had the transpiration on the gas exchange. We had some of those nutrients that are immobile that were having higher levels within the higher airflow rates, and that water use. We would see that more water was used with those higher airflow rates as well. So, and then some of those treatment interactions were hard to explain. I should make some graphs, comparing the different like singular on the airflow versus the light intensity graphs. And then there was cultivar specific responses between these two, although they followed mostly the same trends. Not all cultivars will respond the same. And Dragoon had a lot more tip burn incidents than Casey, and that's just the nature of that cultivar. That's actually why I chose that cultivar versus Casey, because Casey was known not to have as much tip burn, and then Dragoon had a lot, through the literature. And I think it'd be very interesting to even explore higher airflow rates than this. I think that even going even higher could help with it, but I don't know. That could get into some other things as well. Yeah, I want to acknowledge the funding, Project LAMP, UGA and USDA all helped fund it. And Micah Gilbert with Agrify, they donated the LED lights that were used for both of our vertical farms, Chris's and mine. And Dr. Ferrarezi, very instrumental in helped me design this experiment, very helpful throughout, and we appreciate it. Dr. Mark Van Ursel, of course, all the knowledge that he has bestowed on me has been great. And I want to single out Chris Needers and Matthew Housley. I'm now an electrician because of them. I had no idea how to wire any of these lights, and the airflow systems on there, and luckily a lot of it was set up, but we had to do a lot of rewiring, and Chris and Matt really helped me out with that, so I really appreciate it. And Dr. Lombardini, Dr. Bessinger, Dr. Jayla, Samuel, Hannah, and Alan in the lab. We really appreciate all the technical support, and then all of my colleagues for helping harvest. I really appreciate. Some of those were very long harvests, and I appreciate your guys' help with that. Yeah. So thank you. Thank you, everyone.
Excellent, great job. Do we have any questions? Parker, you went from 51 minutes to 40 minutes, so that's
Okay, good.
That's a lot of
Good,
practice.
Yeah, good, yes.
Yeah, so obviously the tip burn was reduced. You had this morphological and the gas exchange data to back that up, that the tip burn was reduced, but I can't remember if it was correlated with increased actual calcium concentrations in the tissue. You kind of went through those macronutrient results pretty quickly, but you would think, if you saw reduced tip burn, that you might see increased calcium tissue concentration.
Yeah, that's
as
what I
well.
would think. And actually, in hindsight, with those, I'll pull up that graph real quick. But in hindsight, actually, with those with sending the plant tissue off, I think that what I should have did was kind of cut the tips of them and send off the tips. Because that's what calcium tipperin is, like it might be okay in the bottom parts of the leaf,
Or just
but
take like the inner part of the canopy or something.
yeah, yeah. So that's why I don't think there was any, yeah, so there was no, with the airflow rates, nothing with calcium
Yeah,
that was
okay.
significant.
Yeah, that's interesting.
And they're actually kind of all over the place. So that was interesting. I was hoping to see
Yeah.
the significance there,
Okay.
but
That's why tipperin research is so complex, because
Yeah.
it is unpredictable.
and I mean, I think that'd be a better option to send like the tips of the leaves off, but I don't know, because that's where you're seeing the deficiency. I guess that's tough to say though.
Yeah, when you need, a certain mass to analyze properly as well.
Yeah.
You couldn't just do the tips, you need at least 6 grams of dry mass. for a
Yeah,
water
yeah,
tag,
that's true.
so that's a decent amount,
Mhm.
Yeah, but that's a good point you bring up, because I thought about the same thing with my tissue results, because like, depending on if it's mobile or immobile, it's gonna store differently in the new versus old leaves, so when you because we just put them all together to hit that requirement, but it kind of dilutes everything, so you're kind of getting a middle ground, so it's less clear to get some of the nuances, so yeah, it's challenging.
Yeah.
It's challenging.
Yeah.
Parker, could you click on the Zoom link
Yeah,
there
I
and
just
perhaps bring
hit
the
it.
panel
You
back?
can just hit escape.
Okay.
Just hit escape.
There we go.
Okay.
And perhaps click on the participants. Do we have any questions over Zoom for Parker? All right, so do we have any?
I've got a quick question about the solar panels.
Okay.
So. Utilizing it as like, a solar. An energy, source, but also as a shade cloth, that sounds like the best of both worlds. But I feel like that can only be maximized in climates that have high light, year round. Cause if you put it into, somewhere where you have limitations on your light in the winter and you install these into a greenhouse, to shade your crops, you're gonna be shading your winter light. So have you heard of anybody actually utilizing this stuff yet, I guess? And, is it removable? Because it sounds like, these are glass panes kind of.
Yeah.
That, you put them in place and they stay in place, for the life, I would assume. And then I didn't know kind of at what concentration those, those sheets were comparable to shade cloth. For example, like that 0.024 concentration, like how much, how much is that actually gonna shade, a crop?
Yeah, here, I'll go ahead and pull this up too. So this is, well, your first point, you do have a great point. I should have touched on this. But yes, it would be, to install the glazing material, it would be favorable to only do it really at the high light levels, like, maybe Arizona or like places down below, like near the equator, would be great to permanently use that. And no, I haven't really seen anyone actually using this commercially yet. There's just been studies going on.
It'd be cool if there was a film,
Yeah,
a
yeah.
removable film.
No, yeah, there absolutely would be. And I know, Mark, like when Dr. Van Ursel and I were talking about this, he was saying even it would probably be best to have them on like a retractable system,
Okay, yeah, cool.
which, might be even more money too, but so that would be cool, having a retractable system. So like even on cloudy days.
Yeah.
Removing them and letting their full light come through the greenhouse. But yeah, that's a great point. And yeah, I think that there needs to be some growth studies really done with these panels. And at the lower concentrations, so like this is the lowest concentration, the red one, and it does block a lot in the blue, but But yeah, I guess it's letting a lot into the red, and I think, red is a little bit more important.
20%. Roughly.
Yeah.
at The
You take
far red.
You could take an integral of those curves to get the actual percentage across that far range.
Today, we're talking about those, these perovskites, are they? How expensive are they, I guess, do they know?
They are expensive. I don't have a number on the top of my head, but they're expensive. Like I knew some USDA guy who was actually working on the, he's working on the Hort farm, and he had some perovskite panels that the USDA funded, but he was using them for very little energy, like to power some fans. So he said that they were very expensive. I have no idea the ballpark, because I don't think these are really even manufactured for commercial use yet. Still a lot of testing going on to get that efficiency up.
They're so expensive that even receiving a grant, Chris, from the university wasn't enough to build larger panels because of the cost of materials
Yeah,
and the equipment to make them.
it costs them, I think, I understand it's about like $1,000, almost $1,000 per one of those like little tiny things. It's crazy. Yeah, so it's
I mean, they're like hyper precise crystals, like
Yeah,
that
they
grown
are,
painstakingly, like nanometer by nanometer.
Yeah.
like-
Yeah.
I was actually looking it up when we were talking, because I had no idea.
Yeah.
But they say one of the limitations is that they don't last very long.
Yeah, that is true, that they do not last.
Yeah. They say that the very first devices, they became non-functional within minutes or hours. So now we're actually
Yeah,
approaching 2 decades, which,
They're
2 decades
making
is
a
pretty
ton
good.
of progress on it now,
good.
and like the efficiency, like the potential efficiency is over 25%. But like when they're trying to commercialize and make bigger ones, they're not able to get that efficiency. So there's still a lot of work to be done, like actually in that solar power sector of just solar power from it. But I think that in the next 20 years, there will be
Or
a
they'll
lot
find
of progress
a
on
new
that.
technology and it'll.
Yeah, it probably will. But like this is I know that perovskite is like the talk of the solar industry. So like there's a lot of resources being put into it. And so I don't,
So maybe
so
they
that's what
engineered in the
that's
room
the
can
best thing they think.
help
would-
developing.
Yes.
All right, so we went a little over time, but I think great discussion. So there's a lot of potential in the combination of both fields, is actually cool to see all of this together, Parker. So thanks again for the presentation. Thanks, everyone.